Well, one moment as I overcome these technical difficulties. All right. Howdy. Howdy. My name is Lauren Gladwell, and I am a second-year PhD student with the Nutrition Department. And today I will be discussing with you epigenetic regulation by a long non-coding RNA miRNA cluster in metabolic dysfunction-associated steatotic liver disease. My focus of research is metabolic dysfunction-associated steatotic liver disease, also referred to as MASLD, along with other metabolic diseases including type 2 diabetes and obesity. Current interventions for these metabolic diseases include exercise, a healthy lifestyle, and a healthy diet. But that's just the tip of the iceberg. Epigenetics, which is the reversible, inheritable changes to gene expression without changing the DNA sequence itself, reveals that there is more to the story. Different epigenetic regulators, such as long non-coding RNAs, microRNAs, and histone modifications, help bridge the gap of the environment and our genome. The environment can interact with our genome through epigenetic mechanisms such as histone acetylation. This can help turn genes on or off under different environmental conditions. These conditions can include nutrition. So we all know that people react differently to diets. But what if I told you that what your parents ate could affect how you respond to a diet? What about your grandparents? What about your great-grandparents? So to investigate the exposure to diet across several generations, we conducted a transgenerational diet study where we looked at different the effects of a healthy diet, so like a chow diet, versus a high-fat, high-fructose diet on different generations through a maternal lineage and the subsequent offspring.
Keep going.
And if I had my lovely image, what we found is that a long non-coding RNA DLU2 actually was significantly downregulated across 3 different generations. And additionally, alongside this decrease, there is an increase in a histone deacetylase, HDAC9, again, wherever DLU2 was decreased. And this prompted the generation of 2 knockout mouse models that we generated. So the MDR mouse model, which was the knockout of the region where DLU2 was, but the also knockout of the microRNA that's embedded within DLU2. And then the MIR model, which was just the knockout of the microRNAs embedded. And what we found is that once we put these mice on chow diet for 10 weeks, the MIR and MDR knockout mouse models had significant weight gain. And what we also saw was that the MIR mice appeared to have more of a MASLD phenotype, whereas the MDR mice presented with more of a fibrotic phenotype. So this really goes to show how the environment can have an impact on our genes, like nutrition, and not only set a precedent for our metabolism, but for subsequent generations to come. Thank you.
Glad that it was fixed. So, hi everyone, I'm Yeon Noh, PhD student in Dr. Yu Xiang's lab in Department of Nutrition. Today, I'm going to share a story of ghrelin signaling in gut-brain axis in aging. Imagine that you have a talk on the stage or having important job interview, but you feel pain and anxiousness, which makes you feel must go to the restroom. Inflammatory bowel disease is a chronic inflammatory disease of gastrointestinal tract. It severely affects patients' quality of life because of the symptoms like abdominal pain, diarrhea, and bloody stool. The incidence of IBD in the elderly is growing, and the pathogenesis of IBD in the elderly is more complex than in younger patients. But within the complex biology of aging, it is still not clearly known how aging contributes to intestinal inflammation. The exact cause of IBD is still not known, but there are many factors affecting the disease, including genetics, diet, microbiome, and immune system. Did you all know that the gastrointestinal tract has the most immune cells in the body? The gut is a huge reservoir of immune cells. Under inflammation, immune cells start releasing the signals to clean up the inflammation. But if they excessively produce the signal, it can damage the tissue, leading to chronic inflammation. Interestingly, those immune cells are highly flexible. They can change their function depending on what directing signal they receive. My research is studying hunger hormone ghrelin as a directing signal. It signals through its receptor, GHSR, in macrophages, which are the first responders in inflammation. We genetically modify young and old mice, deleting ghrelin signaling in the macrophages and induce inflammatory bowel disease. Then we score the disease severity. What we found is deletion of ghrelin signaling in the macrophages significantly reduced the disease symptom. Interestingly, this protective effect was more pronounced in aged mice. Emerging evidences suggest that gut dysbiosis can increase age-associated diseases, including cognitive dysfunction and Alzheimer's disease. In our study, deletion of ghrelin signaling in the macrophages improved memory function in old mice, suggesting that ghrelin has important role in immune cell function, regulating intestinal inflammation, and further contributes to inter-organ communication, gut-brain axis. With this research, hopefully in the future we can leverage the function of ghrelin in prevention and treatment of inflammatory bowel disease as well as age-associated diseases. Thank you.
Hello everyone, my name is Joelle Sfeir. I'm a second year PhD student in the nutrition department, and I'm in Dr. Heidi Vandenbrink's lab. Polycystic ovary syndrome, or PCOS, is one of the most common endocrine disorders affecting 1 in 10 women. Women with PCOS are not only at a higher risk of reproductive dysfunction, but they are also at a greater risk of cardiometabolic diseases such as insulin resistance or type 2 diabetes. We now know that PCOS develops in adolescence and is diagnosed by the presence of irregular menstrual cycles and androgen excess, which is defined as male pattern hair growth, or hirsutism, or biochemical hyperandrogenism. Previous research has established a link between dietary intake and the timing of pubertal milestones, such as menarche or a girl's very first period. We also know that in women with PCOS, lifestyle changes such as diet or exercise can alleviate PCOS symptoms. However, what we do not know is whether dietary intake can contribute to the development of PCOS in the years following menarche. In order to answer this question, we conducted a prospective cohort study in 40 adolescents and study visits occurring at 6, 12, 18, and 24-month post-menarche. In addition to various tests shown to assess androgen excess, we collected 2 24-hour dietary recalls to assess their usual dietary intake, and participants maintained menstrual diaries over the course of the study so that we can determine who developed regular versus irregular menstrual cycles. Dietary data from the first 2 study visits were then transcribed into a specialized nutrition analysis software for adherence to the Mediterranean diet and its components, added sugar intake, and ultra-processed foods. The goal was to determine whether dietary intake during the first post-menarcheal year was associated with the development of PCOS symptoms at the end of the study. Our preliminary analysis revealed that at 6 months post-menarche, greater adherence to a Mediterranean diet and added sugar intake was positively associated with total testosterone, which is a measure of ovarian androgen excess. And red meat intake was negatively associated with free androgen index, which is a measure of systemic androgen bioavailability. Moving to 12 months post-menarche, red meat intake was positively associated with irregular menstrual cycles, and the consumption of non-fat and low-fat dairy was associated with a lower risk of developing PCOS symptoms. These results reveal that dietary intake during the first post-menarcheal year may be associated with a trajectory towards PCOS. Lastly, we plan to expand to South Texas, which is an underserved area with one of the worst health outcomes in the US, to better understand the relationship between social determinants of health and the development of PCOS risk in adolescents. Thank you.